So what I'm going to do is I am going to review why the cell cycle needs to happen in the first place and then go over the cell cycle. So this is explaining the limitations for cell size, because all cells want to do, guys, is grow and then divide. And if they could, they would keep growing until they were ginormous, but unfortunately they can't. And this is why. You got 3 different reasons. Now, the first reason, if you notice in this really cute little cube, what's happening, the pink region represents where nutrients can't reach. Okay, so if the cell is really small, okay, then all the nutrients, so oxygen and glucose, that need to enter the cell and reach at least to the center, right? You can't have any region of the cell that doesn't get nutrients. All of the areas of the cell are going to get nutrients, and that's represented by the fact that there's no pink in that cell. Now, if a cell gets larger, all the nutrients can't get to the center in time. And the reason why is that the rate of diffusion, so going from the molecules, so either glucose or oxygen or whatever they need, they all move at the same rate. So within this time period that this cell got all of its nutrients, this one, that pink region, is starving. It doesn't have the nutrients it needs. Okay, so it's not fast enough. Diffusion, that movement from the cell membrane into the cell, isn't fast enough in order for the cell to get all the nutrients. So that pink region represents starving cell. In this one, here's your cell membrane. Again, nutrients traveling at the same rate. Look how much of the cell doesn't get any nutrients. And so both of these represent cells that wouldn't get enough nutrients, and as a result, they would die. Okay, so that's one limitation. Diffusion. If it's too large of a cell, then the molecules that they need, the nutrients that the cell needs, can't reach all of the cell, and therefore the cell will die. The other explanation is that DNA, the amount of DNA, is limited. And the reason why I really like this analogy, if you have a really big cell or a really big town and it only has one library, not everyone's going to be able to get the books that they need. Just like in a really big cell, you need as much protein to be made as possible, because a big cell needs more protein than a little cell. The problem is you can't get any extra DNA. You're stuck with what you got. Your cells only have a certain amount. They only have one library. And therefore, if a cell gets super big, it's going to overload the DNA, meaning it's going to require way too much of the DNA, and the DNA won't be able to support the amount of protein that's needed to be made, because DNA is required for proteins to be made, and we'll study that later in the year. So if your cell gets too big to the point at which it can't produce as much protein as it needs because it's limited by its amount of DNA, then the cell will die. Okay, so that's a second reason, DNA overload. Too much is being required of the DNA, and it can't live up to that, and so the cell won't get enough protein. The last explanation, so here we're talking about the movement of nutrients into the cell, okay, and the fact that, for example, for these two, it can't reach all of the cell in time. Here it's the exact opposite. We're talking about waste being taken out of the cell. Okay. So remember, the surface area represents the amount of cell membrane that the cell has. And the analogy that I like to use for this is the number of highways, the number of places where the molecules can leave. So for LA, I'm talking about all the different highways you can use to get out of LA. The volume represents the amount of waste that's produced by the cell. So obviously, as the cell gets bigger, there's going to be more waste created. So as our cell gets bigger, as our city gets bigger, this is what's happening. I start with a volume of 100, so 100 people, okay? And the surface area is 1. Now, if I have a volume of 100 and only one highway for people to leave, that's perfectly fine. There won't be any traffic because that's such a small amount of people. Now, when my cell gets bigger, what happens, and what you guys saw in your lab activity with the boxes, volume increases much more quickly with the size of the cell. So as the size of the cell gets bigger, the volume increases way more quickly than the surface area. And so as a result, this is the numbers that you would see. You now suddenly have 1 million people and 3 highways. Both of them increased, but at different rates. So this 1 million people with 3 highways, you're going to have a lot of traffic. Now in the cell, when you're trying to get rid of waste, that's not an option. You can't have that backup. And so that's another thing that's limiting the size, because the larger you get, the more waste you have, but you're not having enough waste for that waste to get out. So those are your 3 limitations to size. So as a result, what happens are different cell cycles. So your cell is going to need to divide. Now, before I get into the cell cycle and what happens specifically, what I wanted to do was talk really quickly about chromosomes. So remember, in prokaryotes, since they're much more simple organisms, they're fine with just this one circular one circular chromosome. Now, what a chromosome is just a package of your DNA. So notice a couple things about the prokaryote. Number 1, it's extremely simple because it has no nucleus and no organelle. So as a result, the chromosome is just free-floating in the prokaryote. Okay? Now in eukaryotes, since we're much more complex, we need way more DNA. So this is how it's organized. You can't just have strands of DNA floating around in your nucleus. So instead what happens as DNA is coiled around proteins, which are called histone proteins. So when the DNA, which is called the double helix, you can see a cool twisted ladder, which we'll talk about later on in this year. When the DNA twists around the histone proteins, it creates a complex, which is called a nucleosome. Now, if you have a long chain of nucleosomes that are unwound, right, they're pretty loose, we call that a chromatin. C-H-R-O-M-A-T-I-N, a very loose, and that's how normally your DNA exists, just kind of free-floating, not tightly wound. That's how it normally exists. Now, once your cell is needing to divide, what it's going to do is it's going to create these cool coils, okay, in order to condense the DNA, and it's going to have supercoils, which are coils of the coils, right? So you can see that coil, it's coiling in on itself, and what that produces are chromosomes. So the only difference between chromosomes and chromatin is that chromosomes are much more compressed and condensed, and chromatin are much more free-floating. All right, let's talk really quickly about the prokaryotic cell cycle because it's really nice and simple. There are 3 stages of any cell cycle. Okay, so you start out with a little cell, which I'm showing right here down at the bottom, and it's going to grow. That's the first stage. So you can see it going from being little to being big. Now, as I talked about in the beginning of the video, it can only grow to a certain size, and then you have all the surface area to volume issues. And so as a result, it's going to need to divide. Now, before it can divide, it needs both of the daughter cells need this chromosome, this DNA. And so what it's going to do is it's going to duplicate its DNA. Okay. That circular DNA is then going to grab onto the cell membrane to make sure that each cell gets a sample, right? Both need a copy of the DNA. And then what happens, as I showed you with the balloon, this fiber is going to go around the cell at the middle. It's going to squeeze until 2 daughter cells are created. And that's how the prokaryotic cell cycle, how that happens, right? You have the growth, then you have DNA duplication, and then you have division. Now, this type of division right here we call binary fission because it's creating 2 binary, and then it's creating this little like indentation, so it's cutting it off. All right. Now, prokaryotic cell cycle, really simple. Let's look at the eukaryotic cell cycle. It's a little bit more complex, but it still has the same 3 things that are happening. So during interphase, and I'll go over this in detail, but during interphase you have that growth that we saw in the prokaryotic, right? You have the growth from here to here, and then you also have the DNA duplication. So you can see it right here in the S phase. Okay? Then during this last little chunk, that's when, and we call that the M phase, that's when you have cell division. Okay, so let's talk about each of them in particular. So this is interphase. Interphase is the longest stage of the cell cycle. Okay, you've got the G1 phase where you have growth and any sort of preparation for DNA replication. You still have the cell still acting like normal, so you have all the cell processes as well. In the second phase, S phase, your DNA is going to be replicated, and we'll go through that in a couple of chapters, how exactly that happens. Then once that's done, once you have that identical copy, then you're going to prep before mitosis, and then you finally have mitosis. Mitosis is right here. In mitosis, what happens is your nucleus is what is divided. So specifically what we're going to be looking at is what happens to the chromosomes, because they're going to be separated. Okay? Here and throughout the rest of the cell cycle, you're still dealing with just one cell. What happens then is in cytokinesis, you have that pinching off of the cytoplasm, and that is the actual separation of the two daughter cells, and so now you have two cells instead of just one. Okay? So we're going to look at mitosis and cytokinesis in detail, but this is all that you need to know about G1, S, and G2 phase. In G1, you've got your cell growth. S phase, your DNA replication. And G2, you're preparing for mitosis. This is interphase. This is the longest, this is the longest part of the cell cycle. But we're going to spend the next couple of days on just mitosis and cytokinesis. And that's it, guys.